thank you so much, everybody, for joining us. We're really excited to continue our webinar series. And this is a series that has been spearheaded by our Dive with Purpose committee group that kind of continues our theme from Columbia Scuba of if you're a scuba diver, let's look for ways that we can put our skills to use in doing some good. And this week, we're really excited to have a guest speaker with us is Josh Farmer. Josh is a biologist with Keys Marine Lab. And I'm sure he'll tell you more about the lab when he gets on and does his time. But the lab is a full service marine field station, and they work with researchers to protect the marine environment around the Keys. And Josh has been with them, I think, 4 or so years. And I'm sure you'll update us on that, Josh. But he's a graduate of UNC Wilmington, so close by to here. So that's awesome. And he's also a PADI Master Scuba Diver trainer, and he's worked in the dive industry as an instructor around the Carolinas and in the Caribbean. So, Josh, we're really excited about having you here and learning more about your topic of coral disease and what's happening down in Florida and the Keys and what you guys are doing to research that. So with that, Josh, I will turn it over to you.
Awesome. Thank you, Lawrence, for the introduction. And thank you all for having me. Thank you, Tim, for setting this up and coordinating. This is a great treat and opportunity to get to share with everybody in the Columbia Scuba Circle of kind of what we're doing down here and what we've been doing for the past couple of years. So nailed it right on the head. My name's Josh Farmer, working at the Keys Marine Lab for the past 4.5 years. I started early 2017. I got my feet in the door right before Irma smacked us. And then I've been here ever since, and I've got a chance to help out on some pretty amazing projects that have come through the lab. And this presentation I'll give you guys is kind of my observations and the roles that we as Keys Marine Lab played in the different projects over the past couple of years. All right, let me share my screen here.
And while Josh is doing that, I'm going to post a link in chat that has a document for some of the links and references that Josh might refer to. So that'll be a resource if you want to go pull that down to save for later.
All right, guys. Can everybody see my screen all right? Good to go? All right.
Yep, looks good.
Perfect. Thank you. All right, so over the next couple minutes here, we're going to talk about Florida Keys coral reefs and the different threats and opportunities that have become apparent for them over the past about 5 years, and then the Keys Marine Lab's role in all of this down here, specifically in regards to the Stoney coral tissue loss disease epidemic, which I'm sure some of you have probably heard of, but we'll go into a little more detail on what it is and how it's affecting our corals down here. All right, so first, let me tell you about the Keys Marine Lab. We're a state-owned field research station, as Lawrence said. We facilitate marine field science and education. We'll have anything from, before COVID times, a group of 20 to 30 students that come with us and go out and do kind of courses out on the water, university level courses for marine biology or marine ecology. And then we also facilitate graduate level research for marine research. This is a group here from Florida State University that was doing, I believe it was oxygen, really sensitive oxygen sensors right next to sediment levels. And that was this contraption here that we were lowering down, or some really cool micro probes that were right above the sediment level. And then we do quite a bit of reef surveys and reef work. This lady here was working with on sponge communities and their kind of the anthropogenic effects on sponge communities. At KML, we are part of the Florida Institute of Oceanography, or FIO, and that is just a statewide resource organization that consists of us in the Keys Marine Lab and then 2 oceanographic research vessels that are located in Tampa and do regular cruises in the Gulf and the Caribbean. So one of the coolest parts that we have, oh, actually, let me backtrack real quick to give you an idea of where we are. Okay, this got a little smaller than I thought it was going to be, but we're about halfway down the island chain at mile marker 68. You guys, if you've been to the Keys, you probably drove right past us, didn't even realize it. We're halfway between Isla Mirada and Marathon. We're that little town with the blinking yellow light and the post office. But it works out perfectly because we're right in the middle of everything. We can get pretty close or pretty quickly up into the Everglades and back into the Florida Bay. And we can be on the reef in just about 5 minutes from our spot. So really cool site. And then the kind of the coolest bit, other than our location in the Florida Keys, is what we have on land, which is this seawater system. It's a pretty state-of-the-art system that pulls water from a seawater well on the property, runs it through a couple of degassing towers and water conditioners and filters. And then it can pump it to these 25 different experimental, what we call tables, seawater tanks. And then in these tanks, we can hold anything from coral, fish, invertebrates, pretty much any sea creature that we would like to study. And then the really cool bit that we can do is this system allows us to manipulate water parameters. So we can, with heaters and chillers up here, we can manipulate the temperature of the water. We can manipulate the pH, and we can actually manipulate the salinity as well. And so that allows us to set up some pretty interesting experimental designs. One of the cooler things that we've done in our system is we did a collaboration with Florida Aquarium and the Coral Restoration Foundation, and we actually did a cropra, or a staghorn coral spawning of their large broodstock corals in our tanks. I guess it wasn't this past year because of COVID, but the 2 years previously. And it was really cool because usually they did this in the field, which is fun in and of itself. You get to dive and all that. But for actual like output of gametes or the spawn, it was much better to do it in the tanks because you don't get all of the little organisms that come to your lights during a night dive. They're not here in our tanks, so they're able to get really clean cultures that the eggs and sperm weren't eaten in, and they actually had a much higher yield that those couple of years where they did it in our tanks.
I think, Josh, you just answered the question. So they actually did better with the spawn in the tanks than in the wild. And did it happen at the same time as it would have out on the reef?
Yes and no, actually. So there's some interesting studies that have come out for that. So for the acroprid spawn that we have with CRF, yes, it happened at the same time. And it was a more prolific spawn, or at least in the fact that we were able to retain more of the gametes. We were able to collect pretty much all of the ones that we found in the tanks. And then it was a pure sample without as much contamination and death of the gametes. So it was a much higher yield in the spawn. And they roughly spawned at the same time. However, we'll go over it a little later, but we did do spawning for Dendrogyra, which is pillar coral, more of a stony coral, not of the weedy branching species. And those had actually been in our tanks for quite a while with the pillar coral rescue, and they did not spawn at the same time as the corals on the reef. And there's actually a recent publication by one of the professors that was doing it with us, Dr. Karen Neely, that hypothesizes that it has something to do with light pollution and it throwing the corals off the lunar cycle that they normally spawn on. They were roughly about 3 days late spawning, which was kind of interesting. But cool, awesome question. And sorry guys, I didn't mention that in the beginning. If you have a question, feel free to throw it in the chat or just shout it out. I'll be glad to answer as we go. Yeah, so, and then as far as KML seawater system goes, they liked it. NSF liked it so much that they decided to help us expand it. So we have a pretty large expansion coming in the fall 2021. We're pretty much tripling the size of the system that we have now, about 75 seawater tables, and we have a large in-ground mesocosm, like swimming pool size, larger than a swimming pool. It's 210,000 gallons that will hopefully be able for some kind of community size experimental procedures as needed. And so all that comes in 2020 or later this year. We're working on it as we go right now. Real quick, pop quiz, so you don't have to listen to me talk forever. I think this is probably overly simplified for this group, but it was just to get you guys thinking and get you to write something in the chat. What is a coral? Is it an animal? Is it a plant? Is it a rock? I didn't sign up for a test. You can ignore it. But if you want to throw in the chat what you're thinking, just to kind of get the discussion going, or if anybody wants to tell me that I'm silly, that's fine too. Awesome, awesome. All three. Somebody's calling me out on my silliness. Awesome. So so far you're all right. There was no wrong answer. Coral is technically an animal, but it has properties of all three. And let's talk about why corals are so freaking cool. Yeah, thanks. Okay, so corals are a very long-lived organism that are specially adapted for low nutrient, high light environments that they live in these beautiful, clear blue, shallow water reef ecosystems. The way that they're able to survive there for such long times and create such large, massive body size colonies is because of this special coral algal symbiosis. And so what they have is these single cellular photosynthetic algae known as Symbiodiniaceae. It's their family name. There's thousands of different species of them. They actually live inside of the coral tissue. So there'll be thousands of those inside of one coral polyp. And then one coral polyp is the single individual unit of a coral colony, genetically speaking or organismally speaking. But this individual polyp then replicates hundreds, thousands of times to create this giant colony that you see here. This is pillar coral, one of our favorite corals down here. We call them unicorns because of how rare they are and how pretty they are and their nice unicorn horns. So this special symbiosis here allows corals to be able to live in a relatively low nutrient environment because the photosynthetic properties of the algae, or the photosynthesis performed by the algae, provides 95% of the metabolic requirements for the coral, up to 95% of the metabolic requirements. The rest is captured by the coral in feeding with those nice tentacles there. As things drift by, it'll grab them and pull them in and deposit it in their mouth. And so the photosynthesis combined with this consumption provides the energy and the molecules needed for something called biomineralization, which is how the corals make these really cool skeletal structures. And actually taking CO2 out of the water, or calcium carbonate really, and they're transforming it into aragonite, which is their skeletal structure that they build underneath themselves over tens, hundreds to even thousands of years that a colony is alive for. So this intricate working is really well adapted for the relatively high light, low nutrient environments, but it has a relatively narrow environmental tolerance for temperature, for light, for depth, which is more of a play on light. And then also for the amount of nutrients that can be provided to this relationship before it breaks down. So all of those things make, even though it allows coral to be a dominant figure in this relatively low nutrient environment, it also leaves it open to some vulnerabilities, especially when you add humans into the mix. So some of the major threats to corals and to causing stress for coral reefs has to do with this breakdown between this relationship between the zooxanthellae, or the Symbiodiniaceae, those algal symbionts, and the coral animal itself. Okay, a major one, the major driver is climate change. Specifically sea surface temperature rising. The average sea surface temperature is rising, and we're getting more and more frequent hot warm water events, which can cause coral bleaching. And just a quick recap, coral bleaching is when the water temperature remains too high for the coral to remain in symbiosis with those algae. So that relationship breaks down, and the coral actually expels the algae from its tissues, and it leaves it this pale white color, because you're basically just seeing through the thin tissue layer straight into the white skeleton underneath. So this coral is actually still alive, but it doesn't have those algae in it, which means it's pretty much starving while it's waiting for those temperatures to go down low enough to be able to get the algae back and be able to photosynthesize and get the nutrients from those algae. So that's one of the probably one of the number one or major threat to coral reefs. There's also a more of a global stressor of ocean acidification, which, as we pump more CO2 in the atmosphere, it's driving seawater or surface water pH levels down, and that breaks down that viral mineralization process, makes it harder for corals to transfer or convert calcium carbonate into the aragonite solution that they use for their skeletons. So that slows down their growth rates and their ability to reproduce at sufficient speeds. And then another major one is coral reefs are really interconnected systems. So anytime we overfish or take a certain member of the system out, it has these cascading effects that can break down the highly intricate system. So any type of overfishing usually leads to blooms and algal coverage on the reef, because a lot of times you're either removing the predators that kept the algal clearers in check, or you're removing the algal eaters themselves. Another one, physical damage. How deep is considered sea surface water? I just saw Lawrence posted that in the chat. So sea surface temperature is usually considered to roughly about 90 to 130 feet. Those first 3 to 4 atmospheres, that's where a majority of your biological processes are happening in the shallower waters. As you get deeper than that, your photosynthesis rates slow down. So it's not monitored as much there, and that's really where you're looking at, you're having those swings. I think for what sea surface that can actually be registered from satellite is going to be 20 feet. So that's more what's being tracked by the 20-foot, or by if you have the coral bleach watch. If you guys get those updates about where corals are bleaching around the world, it's kind of an interesting one. And that's being done by satellite coverage. And I believe they are only able to get a relative estimate for about the first 20 feet of the water. So obviously that doesn't capture thermoclines and things like that. But when you're talking surface temperatures that affect the reefs, you're talking that 90 to 120 foot range is what we would like to look at. So that tool is great for giving you an idea of where it could potentially be happening, but then you have to go out and actually measure in the field to get the stratification of the water. All right, and then the kind of the last couple there, nutrification, or the excess nutrients that we produce as humans from agriculture, wastewater, runoff, fertilizer in your yards, animal waste in your yards. All those things eventually run downhill and into, especially if you live in South Florida, into these lower nutrient environments. This is actually an interesting picture here, pretty striking. It's actually a wastewater treatment outfall. So this is treated wastewater from sewage lines that is actually pumped. This particular one was in Martin County and is actually shut down. But there are still 2 or 3 of these that are operating in South Florida that are just continually dumping treated wastewater, but it is still high in nitrates, ammonia, and phosphorus, which are all limiting nutrients for coral reefs and can cause algal blooms and things like that. So that's definitely still an issue down here in South Florida and the Keys. And then finally, all of these overarching stressors to the coral animal itself can wear down the coral enough to allow it to become susceptible to coral diseases. And so this particular, so this particular disease is stony coral tissue loss disease. It is a relatively recent coral disease found on the reefs here. It was discovered in 2014. And this is it present in a real one of our favorite pillar coral patches here. And this is what we're gonna talk about a little bit more. What is stony coral tissue loss disease? All right. So stony coral tissue loss disease, it was first reported in 2014, Biscayne Bay, Florida. It appears as this really apparent white band of recently dead coral. All right, so when the band is super white like this, it means that the tissue died within the past about day and a half, maybe most 3 days, depending on where you are. And that's what leaves this bleached white color. And then as time goes on, different algae and sediments settle on top of the tissue or on top of the skeleton, and it gets that less pale white color. So the thicker the band, the faster that disease is moving across the corals. So the scary thing about stony, or the big deal about stony coral tissue loss disease, is that it affects more than half of the Caribbean coral species, which is kind of crazy. Like most of the time, different coral diseases that we had studied in the past would affect 1 or 2 species at the most. This one affects over 22 of them, and 22, or it affects over 24 of them, and 22 of them are reef building species. So these larger, massive colonies, like this groove brain coral here. And it can progress super fast with a very high mortality rate. So on the reef where there's our infections present, it infects up to 66 to 100% of the colonies on the reef. And it can kill a colony in as little as a few weeks, in as short as a few weeks, which is insane. Especially when you're talking, some of these colonies are hundreds of years old. And it is rapidly spreading through the Caribbean as we speak. It's currently reported on 15 locations throughout the Caribbean islands. So let me get a little more morbid here and show you one of our friends, baby elephant, which is was a big Ophelia natans that we had on pillar coral forest, which is one of the places that we were monitoring for our dendrogyre or pillar coral rescue monitoring system. And this was kind of just a sentinel of the reef. Every time we went, we'd see him sitting here, and then Cindy, my boss, and who we'll talk about in depth here in a minute, as starting the pillar coral rescue and the pillar coral research, affectionately named the coral baby elephant, because it was the size of a small Volkswagen beetle or a baby elephant. And so this is a kind of a time series of over the years of 2014 through 2016. And I don't know if you guys remember, but 2014, 2015, we had one of the largest 2 consecutive global bleaching events for coral ever. If you guys saw Chasing Coral, that documentary, that's talking about this same bleaching event. So the Keys reefs were definitely affected by that bleaching event. It was pretty much a reef-wide bleaching event. And that happened in September 2014, and then again in September 2015. So baby elephant was perfectly healthy 2014. April, September, it bleached, but it can handle that. It recovered by April of 2015. In September of 2015, it bleached again. This is a picture of it partially starting to recover. You can see the color starting to come back. Eventually, it did recover, but then starting March 2016, the next time we got out to see the colony, we noticed a line of stony coral tissue loss disease rapidly advancing across the colony. And then the next time we went out to monitor the colony in June of 2016, there was 100% mortality of this coral colony. So kind of a little morbid, but kind of give you an idea of how drastic and how quick this disease is able to move through a coral. And then across the reef. It's doing this on multiple corals across the whole reef at the same time. It's just the size of this one was particularly sad / impressive. So that's what we're up against. And unfortunately, one of the corals that was the most affected by this disease epidemic was the pillar coral. And it was actually the first one to get it usually on the reef, and it was the most heavily affected. It is also threatened coral under the Endangered Species Act. And we also advantageously, right here, this is Dr. Cindy Lewis. She's my boss and director at the Keys Marine Lab. Her and her partner, Dr. Karen Neely, they had just spent a whole bunch of time mapping all of the sites for this rare coral across the Keys. So right before all this happened, they had done this massive ground truthing of all of these different sites across the Keys. And then as they noticed that these corals were being highly, highly affected by stony coral tissue loss, they were the kind of the canary in the coal mine on the reef, and the fact that they were threatened, they instituted a pillar coral rescue project. And so they actually ended up collecting over 543 colony fragments. It started out, they were just able to collect pillars of opportunity. They called them POOBs. And those were just pillars that had been knocked over or fallen over during storms. A lot of times they'd have growth on one side of them because they were laying on their side. And so those were collected and treated for if they had any disease, and then conserved in an on-land genetic bank. So at first they were just collecting corals, but then it became apparent, or collecting these pillars of opportunity, but then it became apparent of how deadly and how quickly stony coral tissue loss was specifically to this species and how quickly it was going through the reef. So they started being able to collect colonies that were actively diseased, and then we were actually able to start working on treating them in situ. So kind of give you a recap of the rescue. Over these 5 years, they collected 543 colony fragments. Of there's 123 different genotypes. That means 123 different individuals of corals. Because one of the interesting and cool things they found out about pillar coral is that they're over a site of the size of a football field. There may be 50 or 60 different colonies on these large pillar coral forests, as they called them. But just because they were different colonies didn't mean they were actually different organisms. They were all from the same original organism, and a pillar had broken off during a storm, rolled, created a new colony, and it was just a massive organism across the reef that was all the same original settler. So really cool animal. And once they started to realize that, they realized how few actual different individuals we had on the reef tract here. So during this rescue, KML seawater system became the initial triage, treatment, quarantine for diseased corals. And then once we triaged and treated them, we'd send them up to our genetic bank partners at the Florida Aquarium. There's a lab in Charleston from NOAA that did a lot of the original disease disease treatment trials on them. Frost Science Museum down here in Miami, Moat Marine Lab in Summerlin Key has a few of them. Coral Restoration Foundation and Reef Renewal all have a few genotypes in this genetic bank. And then to give you a little bit of a success story, after collecting all these fragments, we've actually had successful in situ spawning of these corals at both KML and at the Florida Aquarium. The Florida Aquarium actually had a really successful spawning this past year, where they manipulated the light levels and were actually able to cue them to spawn during the middle of the day, which was really awesome because it allowed them to collect more and have more staff on hand to do it. So the idea is to spawn more of these guys, grow them up ex situ, and kind of increase our genetic diversity since it was winnowed so low by this disease epidemic. And I didn't say it, but I had it listed here. Between 2014 and 2020, we lost over 93% of the known pillar coral colonies on the Florida Reef Tract from this disease epidemic. This rescue is what rescued what remains of the Florida Reef Tract pillar corals. So pretty cool. And starting this rescue is what—
Hey, Josh, and this might be what you're saying, is it the same treatment for the pillar coral as it is for some of the boulder corals? Because there was a question in chat about the treatment. Or are they different treatments?
Yes. So actually, perfect segue question into treatment. Is Stoney coral tissue loss disease? We kind of cut our teeth with pillar coral. That was where NOAA Charleston was learning what compounds, there was different studies that have been done in the past with other diseases, with chlorine, with salt, different antibiotics to try and then apply them to the disease site. And so they tried a lot of those on the pillar coral themselves, and eventually what they ended up with was a pre-mixed compound that's base was actually a shea butter base with a few other elements to it. It was made by a dental company. And then that was mixed with an amoxicillin. And that was what was really successful at being applied to the pillar coral. So for treatment, what we would do for pillar coral is we would actually cut away the diseased tissue areas and dead skeleton and apply that paste right to that cut edge to prevent any reinfection. And that was ultimately relatively very effective. And we ended up with about a 90% treatment rate to be able to kind of rescue the corals once we brought them ex situ. And so that's how we learned how to do it ex situ. And that was in partnership with Florida Aquarium, with that NOAA Charleston lab, and then also with Mote Marine Lab and Coral Restoration Foundation. But then going from there, it also affects organisms on the reef, and then it also affects multiple different species. That's a great question, Tim. Tim's asking, does it kill the pathogen or just stop the spread and the pathogen dies off eventually? We don't know. We don't even know what the pathogen is. We know it has a bacterial component that can be knocked down with these gram-negative antibiotics, and those seem to be the most effective against it. But the pathogen could be some sort of virus that then throws the microbial community of the coral out of whack into a dysbiosis, and then that causes the kind of the breakdown of the coral symbiosis and eventual breakdown of the coral health and tissue itself. And I think that's the general idea of what it could be, but we have not been able to find a smoking gun as far as one specific bacterium or virus that causes the outbreak. And I think we're getting to the idea that it's a consortium and kind of a overall breakdown of the coral immune response. But there is still a ton of work being done on that. So what we did know for kind of the practical application of it is that these antibiotics seem to work the best. There was a disease treatment trial that was performed by Dr. Karen Neely, and that's these pictures here. And she tested different disease treatments, the chlorine, the salt, antibiotics, along with different pastes and epoxies, different methodologies, to attempt to see what would work the best across the most different numbers of species. And eventually what she determined was that the antibiotic with the specialty paste was actually what worked the best across species. You can kind of see a success story here. We cut the line or ran the paste line, tissue past it died, but then remained alive on the other side of the line. So that's just one coral, but the majority of species reacted well to that particular treatment. So then with that research that she did here in the seawater systems, we then took it and implemented it into the field. And we first tried it on the endangered species pillar corals. And so one of the things we were doing was creating a trench line, because one of the ideas is that the disease or the microbial infection actually was coming through the interstitial space between the skeleton and the tissue. So by creating that trench line, you cut off that access and you can apply paste into that interstitial space. So we actually use these underwater grinders to cut what we called a fire break line. And then we would pack that line with antibiotic paste to prevent the spread of the disease into hopefully a healthy tissue. We started this on pillar corals at Coffin's Patch, which is right out back of KML, with relatively limited success with pillar coral. Pillar corals, it seemed it was possibly too late for the colonies, or they're just too highly affected that if there's any background disease, they're just a reinfection from another point. However, this methodology has been used relatively successfully on other coral species. With Dr. Karen Neely put together a team at NSU, working in conjunction with Force Blue Team, if you guys have heard of them. I've actually got a pretty interesting video here of them doing some field treatments down at Lukey Reef on some different species of coral. Let's see, let me make sure I share my sound. Not that it's really needed, but here we go. All right, so that gives you kind of a good idea of what they're doing down there. That was at Lukey Reef. They also did it at Molasses. So if you guys are in the Keys and diving, you see those coral tags and those nails in the colonies. Those are actually treated corals, and you can actually take pictures and send them in. The tag itself says exactly where to send them for her research specifically. So you take a picture of the tag so you know where to send the pictures you're taking. But pretty interesting, and it slowed down the progress of disease and conserved some of our more valuable reefs. So Force Blue Team is another not-for-profit organization that is it's its own organization, and they are ex-special forces divers that are kind of given a mission of rehabilitation / a new mission in life to work with their skills in the ocean for ecosystem restoration. They did a relatively large project with the Super Bowl this year, or was it? I guess it was 2 years ago, with 100 Yards of Hope and University of Miami. Where they did a large reef out planning up there with University of Miami. Really cool organization. They stayed with us for a few months while they were doing this work with Dr. Neely. Awesome guys. Definitely support them 100%. All right. So all of that dendro work then led into, oh shoot, FWC, NOAA, Florida Department of Environmental Protection realized there are all these coral species that are being affected by this disease. We should have a genetic rescue for all of them. So then came the Coral Rescue Project. Between 2018 and 2022, they collected almost 2,000 corals. They're targeting all the coral species affected by stony coral tissue loss, but they are taking corals from in front of the disease line. So they're not worrying about treating with diseased corals. They're just taking pre-diseased corals and then bringing them on land as a genetic bank for the Florida Reef Tract corals. KML was initially tagged as the first place for these corals to be held, but they, after the initial holding of about the first 100 or so corals, and 150 corals that we took care of, they've since been moved to other partner locations that are better able to handle large amounts of corals for extended periods of time. And they've actually got a really cool website here that I'm going to see if I can pull up. Maybe. There we go. So this is on your guys' link sheet too. But they've got a really cool ARC dashboard that tells you all the different sites they rescued corals from, when they rescued them, where they're at. The really cool part about this is that there was a whole bunch of AZA zoos and facilities across the country that partnered to hold this genetic stock. So there is you can kind of look and see. I think there's actually one at a zoo or aquarium close to you guys that holds quite a few number of these corals. Yeah, it's just kind of cool to go through. You can see where they're held, how many there are, and what they've been up to. But this is in that link folder that I sent out to you guys. And this was one of the largest collaborations that we did. It was with NOAA, Florida Department of Environmental Protection, FWC/FWC's research arm, FWRI, us, Florida Aquarium, and then all the rest of the AZA zoos and aquariums, a bunch of different universities, University of Miami, NSU, a few others. We're all partners in this organization. Let's see. Sorry, guys.
Josh, if you can see the chat, Mark was asking a question about the tags and were they on just disease corals or
Sorry,
were
guys,
they
COVID roommates. Okay, so that was the coral rescue project. It was kind of an upscaling across all species of the Dendro rescue project. And I think Mark had a question for me here in the chat. Was diving off Key West in the summer after Irma, saw so many tags. Some coral was white, others look normal. Are the tags only for coral needing treatment? Yeah, so they were, the tags were tagged corals for corals that were treated. So if a coral was going to be treated, it was tagged that day, the treatment was applied, and then it was monitored. It is being monitored from then on out. Oh, perfect. And then Tim said it's the Riverbank Zoo in Colombia that is holding the corals close to you guys, and they're actually doing a webinar for you guys in September. All right, one of the last cool projects that's coming. So all of this destruction from stony coral tissue loss disease, and now we're moving into looking at Mission Iconic Reefs. What is that? So it's a multi-year plan put forward by the Florida Keys National Marine Sanctuary. And the idea is that they want to restore with grown corals from both this coral rescue project and from other restoration organizations, 7 of the iconic reefs throughout the Florida Keys. And so they're hoping to make a concentrated effort at these 7 different reefs, restoring up to 15% of the stony coral cover in the first 6 years, which is really large goals even for these concentrated reef areas. It requires quite a bit of scaling up of our restoration efforts, but it's definitely a needed initiative. So they want to start with the restoring stag and elkhorn corals, which they're already relatively good at through different organizations. These are fast-growing species and not affected by stony coral tissue loss. And then they're going to move into restoring star, brain, and pillar corals as it becomes apparent that you're not worried about reinfection at these restoration sites. And then the other cool thing they want to do is start supplementing beneficial benthic invertebrates that have long been lost to these areas, such as long spine sea urchins and the Caribbean king crabs that are prolific algae eaters and would help with coral recruitment and recovery. All right, so in a nutshell, that's Mission Iconic Reefs. I know I'm getting close to my time. I'm sorry, I'm long-winded. But let's talk a little bit about what can you guys do, okay? One, be an ocean advocate. There is a series of online trainings done by CFAN with the Southeast Florida Action Network. And they are excellent. I gave you guys the link to those. There's a whole bunch of coral ID courses, invertebrate ID courses, reef fish ID courses. All of those things are super helpful background information that if you guys are out diving, you can take for us. And then they also have coral disease ID courses to kind of continue your education in marine science and education for diving. Now, obviously, local diving and local debris cleanups. There's the different debris cleanups down here in the Keys, and I'm sure there's some down around by you guys. Things you can do at home limit your personal use of fertilizers and other harmful chemicals that run off from your yard. Everything's connected by water, and we're learning that more and more as we go. So the less you can have of your own personal runoff, the better. If you're in the Keys or if you're in places with these sensitive, or really anywhere, practice safe boating and diving practices to limit your stress on the reefs. And then decontaminate your gear to prevent stony coral tissue loss disease spread. So if you're moving throughout the Keys, or especially if you're moving from the Keys to the dry Tortugas, or if the last place you went diving was the Keys or somewhere else that had stony coral tissue loss, and then you're going to fly to another island that may or may not have it, make sure you decontaminate your gear. I gave you a full set of instructions in the handout I sent, and those are directly from the Florida Sea Fan Network with what to do. But it's a low-grade bleach solution for anything that comes in direct contact with coral, so if you, with your fins or anything like that. And then for the gear that's a little more delicate, so your BCDs, your wetsuits, cameras, and things like that, there is a Quatra ammonia solution that you can buy in the store that does an excellent job, disinfects your gear. And my mind is drawing a blank on it. I'll find it in a second once let me continue here. But all the detailed instructions are in that handout. And that is probably one of the best ways you guys can help prevent that spread of stony coral tissue loss disease. Other things you can do, make your voice heard. So I don't know if you guys have heard about this yet, but the Florida Keys National Marine Sanctuary is creating a new restoration blueprint. They are expanding the size of their spas, or they want to expand the size of their spas and their protected areas in the sanctuary, but they need our help. There's a lot of pushback. So there's going to be a final draft for comment to be released soon, hopefully by the end of the year. COVID has pushed everything back, but it was originally supposed to be out already. But when it comes out, I'm sure Tim can make you guys aware, but check the plan out. You actually can go check it out on their website. I gave you guys the link for that as well. It has a really interesting interactive map of where they want to enlarge and their different plan ideas. Kind of the big takeaway is our reefs here belong to all of us. Everybody has their experiences in the Keys and on the reefs here. So please submit your comments for what the reefs mean to you and how you want to see them protected. It's definitely one of the things that we need to do moving forward in the future to protect the resources that we have here.
And Josh, let me just chime in because I commented on an earlier one, and they actually encourage anybody who goes down there, not just Florida State residents. So we as a diving community and go down there, they want to get our input. And yeah, it's a huge document, but it was really well organized. So you knew what the highlights were, you knew the 3 options, at least in the draft what they're looking at. So it's not going to be a tedious thing to go through like a lot of these studies are. So there you go. So I just want to put a plug in that it's, you know, don't be turned off that it's going to be a huge document.
Perfect. Yes, exactly. Yeah, and if you have any questions, I'll throw my email in the chat. You guys can email me with questions, and I'll get to you as soon as I can. If you have questions specifically about anything for tonight too. But yeah, that's definitely highly important, and I'd love to field any questions as you guys have as you go through it. All right, and then finally, get involved, right? So we already know dive, debris cleanup, and then volunteer dive skills, okay? There's organizations, you guys, I'm sure you guys have heard of Coral Restoration Foundation, excuse me. But then iCare is a new up and coming one. It's Islamorada Conservation Restoration Education. They're doing some really cool like citizen science targeted approaches to this. So you can get in there, you can do fish assessments, you can do coral cover surveys. You can also learn how to help outplant corals, and really help us understand how our restoration practices are helping the ecology of the system. So really cool. I would highly recommend you checking them out. And actually, Dr. Kylie Smith from iCare is going to present on May 20th for this webinar series at 6:30. She's awesome. Highly recommend you checking that one out and hearing her pitch for what they're doing at iCare, because I've been down here for a while. I've seen the restoration practices, and I'm really excited about what she's trying to get off the ground. It's really cool. So definitely take a listen to that and see if you can get in the water and plant some corals for us and help us take our reefs back. And I think that's all I've got. You have questions?
Josh, just as a follow-up too, Josh, I wanted to let you know that we've been with Dr. Smith in December, in February, and we're going again in June.
Awesome.
To work with iCare.
That's great to hear.
Questions for Josh? Well, and one thing I'm gonna do, I always like doing teasers for other stuff going on. And you heard Josh talk about some training and coral surveys, and you might see a lady on your screen, Anna Zamgranese. She is with Sea Grant down in Florida, and they do some similar trainings. A couple of us have recently done her coral ID and disease survey. I wanted to give her a chance to give a little shout out about what her program is, because it is some citizen science stuff on how we can use coral IDs and do the surveys to help feed the scientist people like Josh. So Anna, give you a couple minutes.
Sure. Thanks, Tim. And Josh, great talk. I just finished teaching another webinar, so I joined late, but I was really glad to hear the work you were presenting. And I was actually just down at Summerlin last year, February, right before the pandemic, where I was at your facility for a meeting.
Awesome.
So nice to meet Columbia Scuba folks. I am the Florida Sea Grant Extension Agent in Miami-Dade County, and Tim and I believe Sean from your group both participated in my Stoney coral tissue loss disease observer training. And what that entails is learning about the Stoney coral tissue loss disease outbreak, which sounds like you got a pretty good overview tonight, and then really focuses on teaching coral ID for 11 particular species and skills to characterize coral condition, with the idea that when you being recreational divers, go dive on Florida's coral reef, you take the opportunity to do surveys while you're down there. And so I provide you all the materials. There's an online portal. And at this point, because most of the disease has already progressed through the majority of the reef tract, we're really looking to try and determine how the reef is recovering. So in this sense, reports of no disease are just as important as reports of disease sightings. So it's a 3-hour class. It's free to participate. I've been talking about scheduling a class specifically for Columbia Scuba. So stay tuned for that. And I'll post a couple of links into the chat that you guys can check out. They're just links to blog pieces that I've written. So I hope to maybe work with you all again in the virtual space, and then who knows, maybe I'll get the opportunity to link up with you when you guys come to Florida in June.
Anna, thanks. You've heard some references to the webinar series that we have, or upcoming ones. I just did a link in chat to the shop schedule, so you can maybe save that and look us up later. Josh, you're now part of the Columbia Scuba family, so when I get down there, I'll be giving you this rash guard that you can't see because of virtual background, and it's killing me. Anyway.
Awesome, thank you.